So I've promised to do this for a while, and I finally decided to get around to it. Not too long ago, I graduated with my master's in biotechnology, specializing in CRISPR-Cas9, and I thought I'd like to talk about CRISPR-Cas9 evolution and their structural biology. In a nutshell, everything that I say today, please go and check online for yourself. Go do your own independent research. I am just a lowly master's student. I don't know anything, and I'm just a person from the internet. For all you know, I might be lying. Alright, so let's get started. What are we doing today? We're going to introduce Cas9 in a nutshell. Everything's going to be really basic. Nothing in depth. It's just going to be touch and go. The second thing we'll do is figure out how to find CRISPR-Cas9 systems, just based on their domains and their structures. And we're going to demonstrate that by showing off ISCBs, which is just the ancestor to Cas9, because the best way to find out more about something, in fact anything, is just to look at the ancestors and to look in the past and to use basic computational tools to show off proteins. I created a phylogenetic tree, a sequence-based phylogenetic tree. We'll get around to what I wanted to do, which was a structural phylogeny, but that hit a ton of roadblocks, and I'll explain that later. Everything I'll talk about today can be done at home using online servers. So Motif Finder, you take a protein sequence and compare that against a protein sequence database, and it will find short sections within the protein within the protein sequence that you want to search, and it'll find and allocate and annotate it for you. HHpred is pretty much a sequence to structure based comparison. You'll see that difference later, but in a nutshell, it's more sensitive than Motif Finder. It'll find remote homology based on the sequence, so it'll find a potential structure that may not have been considered to likely fit the protein sequence if you had put it through Motif Finder, for example. So it's just essentially more sensitive. Foldseek is a structure to structure comparison, so we need a PDB or a CIF file, CIF file. We can't use a protein sequence to feed to Foldseek, and it will compare your protein sequence to a massive database of it'll compare your protein structure to a massive database of other protein structures, but it's pretty lenient. It's not very accurate. So you use that for kind of like a quick and dirty structure comparison. And then I'll use TM-Align to do pairwise comparisons of structures, and Chimera X to show the 3D structures of proteins. Then the third thing I'll do is really quickly talk about my troubles putting this presentation together. The reason why it took so long to do this, technically speaking, was because there was a lot more I wanted to do, but we'll go through that later. So, bacteria hate bacteriophages in a nutshell, simply because phages need bacteria to replicate, and it usually ends in the death of the bacteria and the surrounding bacterial colony. Because a phage will inject its genome into the bacteria, and the genes from the phage will hijack host metabolic and replicative proteins to make more of itself. And once the phage finishes putting its progeny together, or once the phage's progeny finishes being put together, they will lyse the bacteria. So they'll break out of the bacteria, killing that bacteria, and then repeating the process with all the other bacteria in its proximity. So that will mean the death of an entire bacterial colony. Over the course of time, there's a kind of arms race going on. The bacteria must defend themselves against the phages. And what you tend to get is these innate defense mechanisms or escape mechanisms that have evolved through natural selection. This includes things like bacteria evolving to have different receptors on their cell surface, which changes whether or not a phage can attach chemically appropriately to a bacteria, and whether it'll just slip off, so to speak. So there's restriction modification, where these proteins within the bacteria will attack non-self DNA. In essence, it'll attack the phage DNA, but not its own DNA. And this is thanks to processes such as methylation, where you take a methyl group and stick it to your DNA, or to the bacteria's DNA, and the proteins will see that methyl group and go, okay, this is bacterial DNA. That methyl group acts as like a tag, so that when it sees any other DNA that doesn't have that tag, it goes and attacks it. The other option is adaptive defense mechanisms, and this is CRISPR-Cas. CRISPR-Cas is an adaptive system in that proteins that attack non-self DNA can store memories of their past infection in host bacterial DNA, and then that memory, so to speak, can be passed on to progeny of that bacteria, because that memory is in the DNA, it's hard-coded in. So it's not quite the same as an innate defense mechanism, because innate mechanisms require natural selection and evolution, but an adaptive mechanism requires the use of a memory bank, a CRISPR array, to put their memories into and then pass on to the progeny. And that will be the topic of today. So CRISPR-Cas stands for clustered regularly interspaced short palindromic repeats, CRISPR-associated proteins. Just to give you a scope of how many Cas systems there are out there, Cas systems have two classes, class 1 and 2. Class 1, types 1, 3, and 4 are multi-subunit CRRNA effectors, and class 2 are types 2, 5, and 6, and these are single CRRNA effectors. Not important for today anyway, it's just to give you a scope of how many there potentially are in the world. And Cas9 is a class 2 type 2b. So it's pretty specific. It goes, there's a phylogenetic tree of all of it. And you'll find that Cas9 is just one of many different Cas systems out there. So how does CRISPR-Cas9 work? When the phage infects a bacteria, proteins from the CRISPR-Cas system will take a chunk of the phage genome and incorporate it as a protospacer, this kind of top sequence there, inside the CRISPR array, which is literally the inheritable memory. You can kind of see how there's one memory here in greens. There's a repeat that separates the next memory, which is blue and red. And there's another repeat here that separates the blue and red from another memory, which is yellow and orange here. In a nutshell, in this CRISPR array, there are 3 memories. From this protospacer, there's the complementary sequence, which is the spacer, and its repeat section, which will be the CRRNA. The CRRNA binds to the tracer RNA, which is usually unique depending on the bacteria and the CRISPR-Cas system. And this tracer RNA, CRRNA bind creates the sgRNA, so the single guide RNA. And then it will attach to the Cas9 nuclease to make the Cas9 effector. So there it is, Cas9 effector. So this Cas9 effector can now go and attack the phage genome by cleaving the phage genome into 2. That's essentially all it does. It just disrupts, breaks the phage genome, it's dead. So all you have to remember is that the CRRNA binds to the tracer RNA to make the guide RNA, and that the Cas9 nuclease is like the nucleotide scissors. So you can think of the H and H and RUVC domain of the Cas9 nuclease as the blades of a scissors. You kind of need both for it to work. You need to have the guide RNA to bring the scissors to a particular part of the genome to be able to cut. Otherwise, there's a section called the PAM domain, or the PAM interacting domain, the PI domain, which will, depending on the Cas system, have a unique sequence such as NGG, which is for SP-Cas9, the super famous one that we'll talk about later. But without this guide RNA, the molecular scissors will literally go around to any nucleotide combination of NGG, which is any nucleotide, ATCG, combined with a G, then another G, and it will just cut there. So it's pretty indiscriminate if you do not have the guide RNA. So as said down here, DNA scissors that are guided to a particular position on the target genome. thanks to the guide RNA. Even though we've known about Cas systems for a really long time, the guide RNA was only just discovered in like 2012, and it was what guided the nuclease over to a particular sequence in the genome, or any genome, to be cleaved. Otherwise, before then it was assumed it was quite random and no one really touched it. It also turns out that the guide RNA can be programmed, so we can change the guide RNA sequence to change where the Cas9 nuclease cuts. Therefore, we can program the guide RNA to get the Cas9 nuclease to cut plant, animal, and human DNA. So, what's fascinating and what I really like to joke about is that we are putting what is innately a bacterial protein into our own cells, or to any cells really, to alter the DNA. Pretty fascinating. So now we get into the repair mechanisms once the double-stranded break has been made. So once the Cas9 nuclease has cut the DNA in, say, a higher organism such as a plant, a human, or an animal, we do have the ability to repair our DNA if it undergoes any kind of break, mismatch, potentially an indel. But in a nutshell, when there is a double-stranded break, your DNA repair mechanism can either be non-homologous end joining or homologous end joining. So non-homologous end joining is generally used for gene knockout or a removal of gene function, simply because once that break has been made, random nucleotide insertions of DNA will be put between the cracks and then fused back together. So it's just random. Whatever that here purple gene used to be is now no longer functional, simply because our body or the cell has just decided, screw it, we're going to shove in these 3 random nucleotides inside just to fill the gap. That's it. Just to fill the gap and to fuse the 2 bits of the DNA back together. So at least we still live, but we're probably missing something. On the other hand, we have homologous end joining, where once the break has been made, you can fit another gene or another specific template of nucleotides into that gap. We call that a donor repair template, and you can generally use this to put in new genes. So it's like, okay, break open the DNA, we want to put in this new gene that makes you glow green, so we fit in the glowing green template in between the cracks, fuse the cracks together, and now you glow green. So I joke about having GFP babies. I probably shouldn't say that. So here's some really quick one slide journal clubs. So for example, in sickle cell disease, chromosome 11, there are point mutations at the beta-globin genes, and these beta-globin genes transcribe express haemoglobin. And this particular form of haemoglobin is badly mutated. So the normal red blood cell, which is round with a little dimple in the middle, is no longer round with a dimple, but now sickled because of the mutated beta-globin genes causing the production of weird haemoglobin. So there's a therapy to kind of cure this. You take blood cell stem cells from a patient. You can use CRISPR-Cas9 to cleave the BCL11A enhancer. It's essentially a regulatory sequence which, when activated when you're born, stops the production of fetal hemoglobin protein. By switching off the BCL11A enhancer, you get the upregulation of fetal hemoglobin protein. In doing so, you are actually not changing the amount of you're not changing the beta-globin mutation, so you're not changing the amount of hemoglobin that causes sickle cell disease. Instead, what happens is that the amount of fetal hemoglobin protein dilutes the amount of hemoglobin that causes the sickle cell disease. Now you infuse these HSPC cells back into the patient, into their bones, and because of that stoichiometric difference of fetal hemoglobin being more than the amount of hemoglobin causing sickle cell disease, you effectively get blood cells that are more likely to become useful again, normal quote-unquote red blood cells that are no longer sickled. In other words, it's just that you have more fetal hemoglobin to produce normal red blood cells than you are to have hemoglobin that causes sickled blood cells. It's just a stoichiometric thing. But Cas9 in a nutshell helped edit those cells. Next. So in Down syndrome, you've got 3 copies of chromosome 21. So this Japanese study took iPS cells, which is just pluripotent stem cells, from a 1-year-old with Down syndrome. And this 1-year-old had 1 paternal and 2 maternal chromosome 21s. They did sequence alignment to identify the 3 chromosomes from each other, so P1, M1, M2, and they found that M1 has a unique target site or a unique sequence that they could send CRISPR-Cas9 to cut. Now they cut it and damaged it enough that the M1 chromosome couldn't repair itself, so that's a dead chromosome, and all that's left is the P1 and M2 for the chromosome 21 group. And technically speaking, this would be a cured child of Down syndrome. And this is just a proof of concept. Once again, I am not going to go into the ethics behind this. So what does CRISPR-Cas9 look like? CRISPR-Cas9 kind of looks like this squiggle over here in reality. So this really simple diagram here is actually this over here. I've changed the H&H color on accident. So this blue, light blue here, H&H, is actually this green domain there. And we can actually show that on Chimera X. Let's go do that. All right. So let's give this a quick spin. We have the RUVC domain here, the H&H back here, this green, the rec domain in purple, which is this huge squiggly thing here, and the PID domain, which is, or the PI domain, PAM interacting domain, which is this red section here. We'll cover the bridge and we'll cover the wedge later, but we're not too interested in what it does per se. We're more interested in just showing you what it looks like. So this is what the CRISPR-Cas9 looks like with the rough amino acid sequence domains for where everything is. And you can find that, quite interestingly, this H and H exists between RUVC 2 and 3, and I think that's something that'll be quite interesting to look at as we start showing off what other proteins kind of look like. So what did I do for fun? Why am I doing this? Because I was bored. To identify Cas systems, the first thing we're going to have to do is look at the ancestors. So the best way to look at ancestors is to generate a phylogenetic tree. I vibe coded and pulled 1254 ISCB, which is the ancestor to Cas9, and Cas9 protein structures from a database. And you'll see that when we show off, or when I show off the tree, it's cheap, easy, and unvetted for similarities. It'll look terrible. If you're a biologist out there, you will probably want to kill me. But, you know, this is quick, dirty, and awful. For safety, I will not be showing any protein sequences. I will also purposefully omit certain bits of information and completely not mention it, simply because I don't want to step on anyone's toes, and I don't want to accidentally be revealing someone else's proprietary information, and I don't want to get sued. Okay. And I ran these sequences, the 1254, on a really simple phylogenetic tree maker. Okay, so it's called ngphylogeny.fr. All the tools I used will be in the description below. So this is the phylogenetic tree I created. This red section here, all of these sequences are ISCB, followed by Cas9s, which is all of this blue section here. What makes it really unique and really interesting, rather, is that you can clearly see that from here to here is all ISCB, and there's this huge jump, so to speak, to a completely other clade here. See that right there? Oops. Right here. That delineates, it's like a borderline that delineates where ISCB is and Cas9 is. So, because we're talking about comparing and looking for the evolution of ISCB to Cas9, we want to compare two proteins at this borderline. I'm going to call them 451 because it's got 451 amino acids, and 1351 because it's got 1351 amino acids. So it's the proteins directly next to each other, separated by this borderline between ISCB and Cas9. Once again, very simple. We're not going to do anything particularly complicated. First thing we're going to do is ask, what is in 451? So we're going to go to Motif Finder. Motif Finder, you just put in the sequence, hit enter, and we'll see what comes up. Here we can see 468538, the topmost value, with the lowest E value, which essentially just means the highest probability of what is there being there, is a guided ISCB. And what is it? Yes, we know it's an ISCB, which is good. The next one, 433801, which covers 0 to about, 2 roughly, to about 179 in the position here, this query of 451. It suspects that there is an RRXRR protein. This is essentially just a overall nomenclature, I guess you could say, for RUVC proteins. So yes, there's an RUVC inside this ISCB. As you can see here, 468538 covers 2 to 319 to here. The program is essentially saying, look, there's likely an ISCB within here. Great. So now we have an RUVC. Great, which means there's one end of the molecular scissors. There's a H&H at 396422. There it is right there. So we can kind of see that under this ISCB domain, there is likely to also be a RUVC and a H&H. And all the way down here at 442735 with pretty, I guess and say it's pretty confident. Here 442735 is saying there's some sort of Cas system that's likely to be within this domain of 141 to 264. This is just one database, the ICBI CTD database. The second database it compares to is the PFAM. So down here, RXRR, right there, comparable to the 433801 from NCBI. They're pretty much agreeing, yeah, there's probably a RUVC domain in here. Yep, RUVC, probably in here somewhere. Remember, back here, RUVC is pretty large and dispersed. There's one at the beginning, kinda. There's kind of one over here. There's kind of one over here. That's fine. It should be comparable because we are talking about ancestors anyway. So it's picked up an RUVC here. H&H over here, pretty comparable to here, 396422. So we can be quite certain that Motif Finder has found us an ISCB which has a Cas system in it that has a H&H and RUVC. Next thing we're going to do, HHpred. What does HHpred tell us? It tells us that the sequence that we've provided, which is just the amino acid sequence, is likely to kind of match to a protein structure with the PDB protein database code of 8CTL. And what's 8CTL? ISCB. And what's ISCB related to? CRISPR. And where in the template? 42495. Sorry, not template. I've highlighted these wrong. Let me quickly change this. PDB, NCTL, ICB CRISPR between 1 and 426. Well, what do we know? Between 1 and 426, somewhere here. So it's pretty much in agreement with the motif finder that there is some kind of ISCB pretty much encompassing the entire protein query of 551 amino acids. Sorry, of 451 amino acids. What's next? ISCB between 1 and 426. So it's like, okay, so there is an ISCB. It is CRISPR-like between 1 and 426. H&H 100 to 323. How can we be so certain? That's the thing. It's not quite the same as the motif finder search between 100 and what is it? Here, sorry. 100 and 323. So it's suggesting that this H&H region is probably pretty big, only with a confidence score of 99.9 and an e-value of something a lot smaller, but it's probably there somewhere. And once again, CRISPR is in there somewhere. So what does HHpred tell us? Well, it tells us there's definitely an ISCB of CRISPR relation. There's a H&H nuclease, and that's pretty much it. Where's the RUVC? I only picked the top 16, and if it's not within my top 20, I'm not considering it. That's all the HHpred tells us. Next, Foldseek. So we go to AlphaFold server, we put in the entire protein sequence, and then we just hit run, right? Just hit run. And it'll produce a very, I would dare to say, mediocre confidence kind of PDB structure or a 3D structure of the amino acid sequence that we can look at. And that's essentially what I'll be showing off later as well. But based off the PDB structure of 451 compared to the structured database on Foldseek, it has given us a match to a H&H domain. Once again, part of that nuclease scissors at 1864242, pretty much based off this model from no cardia, whatever this is. So it's pretty confident with 1 e-value pretty low, it's okay. CRISPR-Cas9 from who? CJE-Cas9. So this will come back to later actually, it's pretty funny. So they reckons that there's a Cas system between 126 to 434 of our 451 query search, and there's an RUVC between 52 and 318. That's not bad. Okay, so what does our protein look like? Well, it looks like this. Based off the really average AlphaFold server fold, we know there's a H&H between 200 amino acids and 350. RUVC between 30 and 370. And we know there's an ISCB, 1 amino acid to about 400 amino acids, and the CRISPR-Cas system likely between 130 AA and 350 AA. So let's visualize that. Let's draw our entire query search, 0 to 451. RUVC between 30 and 370. H&H, remember, exists somewhere within the RUVC. Of 30 to, where's it gone? H&H. 200 to 350. ISCB from 1 to 400. Almost the entire query search is ISCB. And Cas9 is 130 to 350. Not bad. Okay. So let's compare it to a model. So this is what an ISCB looks like. We can see that the RUVC and H&H domains are highly conserved compared to this fellow over here, RUVC, H&H. We desperately need those because if we don't have those, no molecular scissors. So that's essentially all that we're looking for, and that's why the protein databases were constantly being like, look, we've only found H&H and RUVC and ISCB CRISPR-Cas because that's all that's really, really conserved. So if we go ahead and overlap the two proteins using TM-align, which does a structure-to-structure comparison of the two proteins, a pairwise comparison, and then we do a really simple sequence alignment, and I just color in the sequences of 451 based on HCSZ, we can see that the PLMP domain is highly conserved. RUVC, bridge, rec, no, it's technically not really the rec domain. It's too small to be a rec domain, but it's there. RUVC, H and H, WED, and TI. You can pretty much see it's in a very similar fold. It looks very the same, which is good. So let's show that in 3D space. There she is. It's a bit disgusting. There we go. So I have purposely made 451 a little bit more translucent so you can kind of see through it. So you can kind of see through it and you can see where it overlaps. And if we zoom in, you can essentially see the RUVC, these beta sheets are highly conserved. The red and the blue from the HCSZ, they're pretty much in the same fold, so it looks really good. H and H, you can see how the H and H of the HCSZ is up here, but our 451 H and H is kind of dangling down here for some reason, and I would chalk that up to the AlphaFold server being a little bit subadequate at protein folding. But generally speaking, it shouldn't be too much of a problem because, like a molecular scissors, it opens and closes. To give you some context by what I mean by opens and closes, HCSZ and a lot of other proteins that have undergone cryo-electron microscopy, which is the only way to really get a good look at what these proteins look like, it's just high-resolution microscopy. A lot of these proteins are constantly in movement, if not all proteins are constantly in movement. So to stop them from kind of jittering and shaking all over the place, you need to give it something to attach to. So HCSZ was given a DNA template to kind of just hold onto so you can take a picture of it. Otherwise it'll just keep moving and it'll be really difficult to take a picture of. And although I don't show it here, the protein the DNA template runs through here pretty much, which allows this H and H domain to come up and attach to the DNA sequence or to the DNA template. Just something to attach to. Whereas 541 doesn't have that luxury because we've only just fed the protein sequence to a server without any kind of DNA template to compare to or for it to adhere to. So the H and H just kind of sags down to here, probably. That's just my theory anyways. You can also see how the TI domain is highly conserved. This TI domain is important because that's where the whole ISCB system uses to attach itself to the unique quote-unquote PI domain, the PAM domain, which I was talking about, the NGG, where if the CRISPR-Cas system, in this case the ISCB, didn't have a guide to show where the nuclease should go and cut, it'll literally just go and cut at where the PID, it'll literally go and cut at where the NGG domain is, and it'll begin by attaching to the PID domain. Same thing with ISCBs. Instead of a PID, they have a TI, or a TAM interacting domain. Same logic. So you can see how well conserved these folds are there. Some reason, once again, because of the DNA template, probably, this kind of fold here for 451 in red compared to the orange fold here. The orange fold is the wedge domain. I suspect this red fold here is also the wedge domain of the ISCB. ISCB 451, sorry. Not in the same fold, but that's okay. What's important to remember in a nutshell, RUVC, H and H, they're all there. So, moving on. Let's talk about 1351. This is our CRISPR-Cas system that exists on the other side of the border of ISCB to CRISPR-Cas. 442735 in the in the query search query is a CRISPR-Cas system. Lo and behold, what did we expect? 433172 down here, H&H endonuclease, 436565 RUVC. Great. So within this Cas system, there is a H&H there, and there is an RUVC. It's only captured, say, maybe like chunk of the RUVC. Once again, RUVC is broken up into 3 chunks, which sequentially they are separate from each other. So RUVC there. H&H down here in the PFAM world, I guess you could say, PFAM database world. H&H down here, it matches with NCBI. RUVC3, so it's decided to be more specific, and it's found the RUVC3 subunit. There are 3 subunits, so it's found the 3rd subunit. And another Cas9 alpha helical lobe domain. Yeah, whatever. It's just a Cas9. So what do we know so far? It's a Cas9 with an RUVC and a H&H. That's all we've got. What's in 1351? So we can see here. It's got a Cas effector. Yep, between 2 and 1280. Definitely another Cas at 1 to 1284. This time pertaining to a protein database match of 6kC8. That's fine. H&H is huge. What? The query search is between 1 and 1280. Whatever. It assumes there's a H&H in there somewhere. It's decided to pick up that the sequence somehow links to SA-Cas9. Whatever, it's just a different type of Cas9 between 1 and 1280 and the H&H domain 1282. Sorry, what does HHpred tell us? 1351 is definitely a Cas protein. It has a H&H nuclease. That's pretty much it. What does Foldseek tell us? Oh, would you look at that? Once again, CRISPR-Cas, but in relation to CJE-Cas9, something we'll talk about later. Search query between 3 and 1282. Not bad. H&H, there it is. Another H&H, there it is. So all they've told us is that there's likely going to be a H&H domain within this CRISPR-Cas system that matches to CJE-Cas9. Once again, no RUVC detected, which is kind of weird. What does 1351 look like? Probs look like based on the really poor AlphaFold server. Well, it looks like this. Let's put it up on Chimera X. Alright. So we're going to zoom in and have a quick look. We can see there's a bridge helix right there, which would have been gray in our templates, but we'll talk about that later. RUVC, H&H tucked somewhere back there, here-ish. This is our rec domain. PI domain, somewhere here with the wedge, somewhere here. And we'll see later, which I was a bit confused by when I saw this. What the hell is this? And, you know, that's what you get for using AlphaFold server. You get things like this. What is this growth? What is it? I don't know what it is. Here's the thing, you can actually search what this growth might be on a site called Progress. But once again, with any training model and anything that you feed it, the smaller your sequence, the harder it is to match to anything. So generally speaking, if I were to give this domain over to to Progress, it's very unlikely it'll tell me what kind of domain it is with any kind of accuracy, because it's just too small. How big is this? 1000. Oh, it's at the end, 1351. Yep. all the way to what? What's this? 1286. Huh. Whatever. There you have it. That's 1351. You know what that might be? There might be some sort of like leftover from the PLMP, but it doesn't seem to be the case. Whatever. I'm not even going to think about it. I'm not going to think about it. Not right now. I've already done my thinking. I'm not going to think anymore. That's what I thought it was. Originally, I thought it was some sort of leftover from the PLMP, but the alignment doesn't make sense. I'll show you why. So just to give you a quick idea, the PLMP from ISCB has been known to do nothing. It just does nothing. It's just a weird protein section, a protein domain that exists to be a characteristic tag for calling ISCBs ISCBs. And the fact that it exists in the same kind of fold, I'll show you later actually. I wonder if I do an overlap. I don't remember if I do an overlap. If I don't, I'll do one now. Anyways, H and H between 450 amino acids and 900 amino acids, RUVC, 76AA to 911AA, CRISPR-Cas. And so you can see how you can visualize the information that you've got based off everything that we've done so far, based off the very limited information that we've got so far. And so let's compare SP-Cas9 to 1351, and what we get. Once again, we've got this weird growth being weird and not conforming to SP-Cas9, but that's fine. Let's bring up Chimera X. Oh yes, I use a Chimera MM, that's why. Okay, that's not good. So originally I wanted to use TM align because TM align is, once I said it before, is more accurate. But sometimes because TM align is server based, so if the server goes down, you won't be able to use it, which kind of sucks really, which is something else I'd like to talk about later. So bridge domain, very much conserved. RUVC, I probably should have chosen a different color for RUVC and for 1351, but you can see how the arrows, dare I say, the beta sheets are pretty much in the same spot. Here, 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 there and there, there and there. Pretty good, not bad. PID domain, you can see those folds are also pretty similar here, there. It's not great, to be honest. Could be better. It's a bit stray, really, because, let's see, got a lot of alpha helixes here from the model 8CSZ, our SP-Cas9. Sorry, from our model 7S4X, our SP-Cas9. That's not really aligning to the to 1351. Probably not too much of a difference, to be honest, but you know, little bits of difference make a big difference in my opinion, especially when it comes to 3D conformational structures and protein characterization. There's the rect domain, highly conserved. You can see how the blue kind of almost merges perfectly with the purple. H and H, ugly, very, very ugly, but it's kind of there, the green matching with the blue. Not perfect, but pretty similar. There you go. So if we compare the SP-Cas9 to 1351, you can kind of see how, as I've said before, bridge helix, RUVC is very similar, H and H very similar, TAM domain, wedge, there it is, the PI domain, there it is, and that weird growth that doesn't really match to anything, it's just kind of there. Once again, this is a very simple kind of representation of a sequence alignment. There are tons of gaps that I've decided to omit simply because those gaps are like, what, 10 amino acids in size. If it were up to me in a more professional setting, I would definitely kind of highlight that. But what you would see is these kind of what I like to show off as blank spaces in between the query 1351 compared to SP-Cas9, and you'll see tons of blanks. But in general, it's pretty damn well conserved. So what am I showing off now? Yes, 1351 compared to 451, the grand finale. Is it the grand finale? I'm not too sure. Actually, no, it's not. Once again, everything I'm doing is very low effort. There's like literally no effort put into this. Nothing I have done here is all that special per se. So, big problem that I want to quickly talk about, really quickly about Matchmaker in Chimera X. Can you see how this bridge helix isn't aligned with this bridge helix? Even technically speaking, it can be. But for some weird reason, Chimera X has decided it really wants to match RUVC together. Essentially what that means is that Chimera X has a tendency to base accuracy on the largest domains of two proteins that are most related to each other, and it'll combine them that way. And I have a diagram to demonstrate that later. But because of my choice of cheap and easy, this is what I get. The RUVC looks really good, but look at this bridge helix. It could be overlapping, but Chimera X has chosen to favor the bridge helix. Has chosen to favor the RUVC. Once again, a bit weird. H and H is down here. Our H and H for our ISCB 451 is down here. H and H for, let's have a look. 1351 is somewhere here. So once again, droopy 451, very, very droopy. PID TAM right there, not well conserved. And it's not well conserved because the TI or the TID, the TAM interacting domain of the ISCB, evolves to become the PID. So that's pretty important to remember. So it's not well conserved, but you can kind of see how the structure is very kind of similar. You get these beta sheets, you get beta sheets down here as well. In function-wise, very similar, structural-wise, very different. That's something to remember to a degree, is that just because a sequence looks the same as another sequence doesn't necessarily mean that they look the same in 3D space. And you can't compare function based on sequence. It's better to compare function based on structure, because if you have something that looks the same as something else, it's likely it performs the exact same as something else. If you take a lock and two keys, one is made of gold and one is made of silver, simply because of its structural sequential difference, it doesn't really matter just based on whether or not it's gold or silver if it can open the lock. What matters is if the keys are of the exact same size and of the exact same teeth or whatever for that lock. So structure is more important than sequence. And I'll talk about that some more later. I keep saying that. Why do I keep saying that? Because I will talk about it some more later. Except for the bridge helix part, because that's just protein domains that we don't really need to cover today. But most importantly, something to remember to you future biologists out there, or to you to kind of verify and clarify my own understanding, is that structure matters more than sequence, because structure can dictate, is a better indicator of function. So, quick comparison. You can see how the rect domain of 1351 is substantially larger than the rect domain of 451. In fact, I would hazard to even call 451's rect domain a rect domain. I think in some cases they just call it the linker domain because it's just so small and insignificant. It's just kind of there. That's for another reason, but we won't go over that today. The PLMP domain kind of disappears from this Cas9 like 1351, and the PID evolved from the TID, and that's pretty much it, really. And there's still this weird kind of sequence here that's not really this weird kind of tumor of alpha helixes that doesn't really match to anything. In conclusion, what do we know so far? Based on what we've done so far, what do we know about ISCB and Cas9 evolutionary pathway? Is that the ISCB loses the PLMP as it becomes a Cas system. The ISCB's rec domain becomes larger and becomes a fully fledged rec domain when it becomes a Cas9 system. And that the ISCB TI, the TAM interacting domain, becomes larger and becomes a PAM interacting domain. So just to kind of summarize all of it and just compare the two model systems that we've been talking about today, the HCSZ and the SB-Cas9, these ones are like 100% verified, exactly what they are, exactly in the fold that they should be. So what we can see here, what's conserved between the ICB and Cas9 is the RUVC here, right? The H&H, the green section, you can see how nicely the green section kind of overlaps in the back over there. The PI and TI domains, so there, you see that brown and the red overlap very nicely. Yeah, let's just show it off. Why don't we show it off? And that the ISCB eventually evolves to get a larger rec domain, and the ISCB loses the PLMP. Let's just show this off really quickly. That's your bridge helix. PLMP, a tumor, now kind of lost. RUVC, look at those folds, baby. Look at those folds. You can kind of see how the ISCB, I'm pretty sure, is lightened out. Those beta sheets overlap very nicely. Alpha helixes very nicely as well. Rec domain, from something very small. Sorry, ISCB is not translucent. From something very small becomes something very big. There, all right. H and H, pretty well conserved. Pretty much in the same fold, especially kind of like there. Oops, sorry. Whether or not it matches doesn't really matter per se. It's more about whether or not most of the structures are there. Because at the end of the day, you know, as I said before, they need something to hold it still, and these are all independent studies from each other. They should be, if I'm not mistaken. If I remember correctly. The TI domain, significantly smaller, becomes a significantly bigger PI domain there. So let's do the last thing. Let's talk about all the other ISCBs I sampled. So from across the entire phylogenetic sequence-based tree, I took from the beginning of the ISCB, from the middle, and from the end. And we get in yellow here, 425, in green here, 493, and in red, 451 amino acid, right? They all look pretty similar at face value. That's just how it is. Welcome to evolution. Same thing with the Cas9 section. We took from the blue, light blue, beginning, middle, end, and we can see how these are significantly larger than the ISCBs. And they all look quite similar at face value as well. And because of this, we can make a really kind of simple kind of like timeline of evolution, where we can see that the size of the protein gets larger. We can see that the PLMP kind of drops off, and the rec domain becomes much larger. But what's conserved is the H&H and RUVC, because we need that for nuclease activity. Going forward, from here, essentially, all you need to do is wet lab testing, see if the ISCBs work, blah, 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 see if the Cas9s, if you've picked work in bacteria. Not going to go into that, because that's when it gets really dry and boring. There's no pictures, literally no pictures. Well, there are pictures. It's just pictures of bacteria living or dying. That's it. So I'm going to end it there. So problems I faced making this presentation. I spent a stupid amount of time trying to get AlphaFold to run on my PC locally. So if you're a computational person or bioinformatician, you know for certain that I am a special kind of person for trying. I wanted to make a structural phylogeny of 1,254 ISCBs to Cas9 sequences, and fold all the structures myself. In a nutshell, you need to download the database and compare all the structures that you want, the 1254, to the database. How big is this database, you wonder? Well, the ones I downloaded equated to 2.76 terabytes of memory. I am trying to do a multi-sequence alignment, an MSA, of 1254 sequences to 2.76 terabytes of sequences and protein sequences. Sorry, protein sequences and eventually protein structure sequences. This is not possible on 16 gigabytes of VRAM and 64 gigs of RAM. It takes too long. So I broke up the multi-sequence alignment section into 59 shards, kind of components so that my computer can handle it appropriately. I had to cap the amount of RAM being used at 48 gigs of RAM rather than the whole 64, because during the compilation stage of the multi-sequence alignments, because I've broken them up, and it's just for one shard of the 59, it will use up all the RAM and have an out of memory error and blue screen my PC, and it crashes the entire thing. And I've been trying to do this for about 2.5 weeks, for 2.5 weeks. Now, every time I wanted to compute one shard, it would take 1 day, 24 hours. So, no, I'm not doing this. This is crazy. It's insane, and it will wear down my hardware, so I'm not doing it. The other thing I wanted to mention was the reason I wanted to do this was because AlphaFold servers, I'm not talking about the AlphaFold server per se, I'm talking about CollabFold Online, a much more accurate server, dare I say, web server, only allows a few thousand sequences to be folded every single day. In hindsight, I probably should have just used that for the 3 proteins that I wanted to fold. It was a lot faster as well, but I thought, you know what, let's just do it myself. Anyway, I tried to send a fold job of 1254 sequences to that server, and I got a timeout pretty damn quickly. It realized, wait a minute, you're trying to fold 1254 sequences, which would take up pretty much all of our computational space and all of our memory within less than a day before we can even clear it or clear the cache. So I hope they don't put my IP on some sort of blacklist. Anyway, the other issue that I had was the DALI alignment server was down. And I would much have preferred to do most of my alignments on DALI Align. But what's happened also is that it started working just as I put together this presentation at the very last minute. So I'll show you what that looks like later. But the DALI alignment allows for structure-to-structure database comparison and is significantly more sensitive than Foldseek. Foldseek is like, whatever, man, skim every database and somewhat compare your protein to the other proteins. I'll show you just what I mean by more accurate. So with DALI 5.4, with DALI, when I sequence searched or structure searched 451, it actually figured out that it had a pretty close comparison to 8CSZ, which is the model protein that I was using. I was like, damn, that's the kind of accuracy we're talking about. So this is why I wanted to use DALI very early on. You can see it also finds a H&H. It even finds an sgRNA at the bottom there at 16 here. That's crazy. Like, it knew immediately like what we were searching for, which is an ISCB, which is CRISPR associated with a H&H. You probably also want to check for sgRNA, and it found one. Holy crap. Where? In chain B of 7Z4D. That's crazy. Does H&H do that? Let me check. Can't quite remember off the top of my head. Yeah, it kind of does. That's a chain number there, but you see with HHpred, you can see it's just not as accurate. What, 1426? DALI. RMSD, yeah, it doesn't really show, but still, it figured out that I was using HCSZ as my own model because it just looked like that. It looked like it matched really well. It's pretty sick, actually. And here, for 1351 H&H, CRISPR-Cas. So I much prefer DALI, because DALI also gives me a Z score. That's what I wanted to talk about. Does H&H give me a Z score? I don't quite remember off the top of my head. No, it doesn't. It gives me an E value, which is the equivalent of a probability value and a probability percentage. Yeah, no. This is much better. It gives me an RMSD. So RMSD is how many angstroms difference. So it's kind of like a measurement of difference at a minute molecular level between my protein search and the protein that has identified with. The alignment, length of alignment is like how closely associated based on how many amino acids. NRES, nucleotide, something I can't quite remember off the top of my head, doesn't matter. But that Z score, the higher the Z score, the better, meaning more aligned. Let's see what else is there. Percentage ID doesn't really matter, because it's looking for an overall like amino acid to amino acid match. So A to A, B to B, C to C kind of thing. Molecule. Yeah, no, I just much prefer DALI align over HHpred. And if it were up to me, I would probably skip Foldseek for DALI. That's what I would replace it with. I'll still probably go with HHpred because it does look for remote homology based on the sequence rather than the structure. So it's kind of like it's a good kind of end test for a sequence just to see what structure it could possibly compare to. So I'd still keep HHpred, but I'd go in the order of motif finder, HHpred, skip Foldseek, and input DALI instead. Then do TM align to get the protein structures. That's what I would do. Pairwise protein structures, sorry, for comparison between models and the query protein. That's for TM align. That's what I would do. So motif, HHpred, DALI, TM align. So let's quickly talk about Chimera X matchmaker versus TM align. So if you have protein A and protein B, two different proteins with a similar kind of circle at the bottom, right? They're just shapes. Chimera X will compare preferably the two circles first and say, look, high similarity. It'll give a really low RMSD. So once again, low RMSD means low angstrom, means closer fit, just simply based on the overlapping circles. And this gives an inaccurate conclusion over the about the overall fit of a protein to another protein. Alignment, sorry. TM align will more likely overlap as much of the protein as possible, and simply because that the triangle of the red and the arrow of the blue protein doesn't match perfectly, it'll give a lower similarity and give a more accurate conclusion of what a more accurate visual representation, I guess, and structural comparison of the red protein to the blue protein. So I don't like Chimera X matchmaker. I much prefer TM align, which is why I use that. Now, before I end, the funny thing. Random fact about this presentation. The randomized CBs and Cas9s just so happen to be all related to each other. They all have a common ancestor of CJE Cas9. So even though I vibe coded and quite literally, quote unquote, instructed for a random collection of ISCBs and Cas systems, the reason why we started getting things like, where is it? This here in Foldseek that said the CRISPR associated protein that it matches to 541 is CJE-Cas9, and here as well, CJE-Cas9 for 1351, is because it turns out at the very end of the phylogenetic tree, the most evolved point of the phylogenetic tree, is this really flat part there. And this flat part here implies that all those sequences are very, very related to each other. That there's no sequence that's particularly special from another one by poking its head out more than all the others. So it's all just flat. Because that entire flat domain or flat region there are all CJE-Cas9 sequences from different studies. That's why it's so similar, and that's why everything from across the board, from all of its ancestors, from all the previous Cas9s all the way to the ISCB, all match quite closely with CJE-Cas9. So that's pretty funny. That's it, I think. That's all I really wanted to cover. Thank you for listening. Number one. Number two, please take everything I say with a massive grain of salt. I am just a person on the internet. Whether or not I do have a master's degree does not matter. I am a random nobody who will never prove anything to you other than what I've said so far. So please go and check online for everything to verify everything that I've said. Is the best thing to do for yourself. A lot of the world is moving towards structural biology, bioinformatics and structural biology, because we can do so much with very easily available tools that are just online. Like everything I've done thus far, all online, completely free, with a reasonable degree of accuracy. And I would dare to argue that all the work I've done for this presentation per se is the basics of a lot of the beginnings of any kind of biological research or research into proteins and protein function. Because by using these bioinformatic tools, we can really quickly sift through what works and what doesn't work just based off protein structure, catalytic residues, which, these softwares can show and can program and demonstrate exist. And we can very quickly sift through what works and doesn't work. So it's amazing. If I were to argue how much of this protein modeling stuff was of my master's degree, I would say it was a solid third or more. So I hope this has been mildly educational. I hope this gives you a greater understanding and appreciation of where we are going with biological sciences. And if there's anything that we want to be doing prior to wet lab work, it'll be quickly and confidently identifying protein structures. Once again, like the key metaphor that I kind of used, it doesn't matter what color the key is, like the sequence or what the key is made out of. What matters is the structure of the key, and whether or not it has the same teeth, the same length, whatever, for that lock. Same kind of logic. My name's Mayhem Star, and I'll be uploading more horror videos soon. And I'll see you next time. Bye-bye. Bye.